Welcome, everyone, to this first VHP4Safety ONTOX Early-Stage Researcher webinar series, which will be about research output management strategies for animal-free risk assessments, about Biostudies and ToxTempAssistant, and their implementation in ONTOX and VHP4Safety. So first of all, this webinar will be recorded. The recording has already started, actually. And that's so that people that cannot join are able to find it later on the YouTube channel or YouTube channels. So first, I'd like to introduce a bit the two projects, so ONTOX and VHP4Safety, and explain a bit the reason for this webinar series. ONTOX is a European research project that I received from the European Union Horizon 2020 Research and Innovation Program, and is coordinated by the Vrije Universiteit Brussel, and is running since May 2021 and until April next year. And VHP4Safety, on the other hand, is a Dutch research project, which is funded by the Dutch Research Agenda and coordinated by Utrecht Universiteit, Utrecht University of Applied Sciences, and the RIVM, the Dutch Research Institute for Public Health, and is running a bit in the same timelines as ONTOX since June 2021 and until May next year. And both these projects have a common goal of advancing human risk assessment without reducing the use of animals. So the first meeting for cooperation happened in October 2022, and 6 collaboration groups based on common themes in the 2 projects were formed in 2023, and a memorandum of understanding was signed in February last year. So these collaboration groups are research output management, which we'll see some examples of today, stakeholder involvement, quantitative AOPs, chemical selection, IT platform, and risk assessment. And the goal of this webinar series is that each webinar will be focused on one collaboration group and is an opportunity for early stage researchers to present their work. So it's an opportunity both to showcase the collaboration between VHP4Safety and ONTOX, but also to promote our early stage researchers. And this will happen about every 2 months. So our aim is to have all 6 webinars this year. So keep your eyes peeled for the next one, which will be also about stakeholder involvement. And this is organized in collaboration with Aspis Academy. So I am actually both a representative of ONTOX and of Aspis Academy, and Annick de Winter, my co-organizer, is a representative from VHP4Safety. And Aspis Academy, the reason we are involved is because we are a network for early-stage researchers developing new approach methodologies in toxicology. So we're all about promoting the networking, providing training, providing the research of early-stage researchers, so that the new regeneration of scientists can shine. And the Aspis is the 3 Horizon 2020 research projects, so ONTOX, Risk Hunter, and Precision Tox. So our training program, we give in-person training at Aspis Open Symposium, but also during project meetings. We have a summer school, and we give workshops about soft skills like science communication, presenting, funding, but also about some hard skills. So AI and risk assessment, methods development for regulatory applications, etc. We have a whole host of people in Aspis Academy core group. One of them who is actually presenting today, Eliška. And we also have, we have researchers representative of all 3 projects, and also project managers and coordinators from all 3 projects are in the Aspis Academy board. And if you are interested to join, if you are a young researcher or maybe not so young, that also doesn't matter, you can find the QR code to the LinkedIn group, our website, and our email address. Now, without further ado, let's move on to the actual subject of the webinar, the research output management strategies. So we will have 4 speakers today. I will give a very brief intro of them, and they can speak a bit more about themselves if they want to. First, we have Daniel Jenne, which is an associate professor in toxicogenomics at Maastricht University. We'll have Marcia Verheijen, postdoc researchers in translational genomics at Maastricht University as well. Eliška Kučovská, who is a postdoctoral researcher in developmental neurotoxicology at the IUF Leibniz Research Institute for Environmental Medicine in Düsseldorf. and Jente Houweling, who is a data steward and PhD candidate at the RIVM and also affiliated with Maastricht University. And for now, I will stop sharing and I will give the word to Daniel, who will introduce our topic.
Thank you very much for the introduction. I will share my slides. If I'm correct, this is in presenter mode.
Yes.
Perfect. So it's a pity that I can't see anybody who is online. I did see a few names, so welcome everybody. So I'm going to focus and set the stage basically for the other three speakers, and I will be talking about research output management strategies, focusing on the 5 Ws. But before I do that, let me first shortly introduce myself a little bit, because I'm one of the young researchers, young at heart still. And I started in 2007. You can see me here in the picture. In the field of toxicology, basically toxicogenomics. And in this picture, I've shown this some time ago, also at the VHP4Safety hackathon. And this depicts basically the work that I've been involved in, moving on in different projects, European national projects, where it was crucial to collect toxicogenomics data most of the time, transcriptomics data, and what was needed for that. So basically this sets the stage a little bit. So there were developments on templates, developments on databases, and learning from that is that reusing what is already out there makes a lot of sense. Well, having said that, let's move on to the 5 Ws. So why is research output management important? For who? What is it? And what needs to be done? Where store data, and so on. When should you apply this type of management, and why? Well, the who, that's basically all of you. I summarize it as the researcher. And it doesn't matter whether you're a PhD candidate, a lab technician, student, professor, postdoc, or doesn't matter, just give it a name. Anybody that is involved in research and creating research output should have good management of it. Also applies to whether you're from academia, industry, clinic, or any other type of business. You need to do the work. You also need to make sure that your data and tools and whatever you create is captured in a good way. So let's focus on the what a little bit. So if you have a typical experiment in the lab, in vitro experiment, we're talking about NAMs. So we have a petri dish with cells, expose it to some compounds, then we get some samples out of that. These samples, they need to be analyzed. Analyze different methods, and that will result in some data. And these data can be then further analyzed, data analysis as such, and that will result in your final result. So put now the names underneath it. So what do you then capture in the end in your data management? Basically, it's everything from the start till the end. And I put a question mark behind final results because your results might lead to new research and in the end are then not yet final. So all these aspects, and this figure will come back, are important to capture. So you need to have a good description of your experiment, what samples you collect, what you do with the samples, how you analyze them, how your raw data will look like, and what you do then next, what tools you use for the further data analysis, and how you're going to present your final results. And this has been also nicely summarized a bit in this overview from the paper that is listed here. And that means that you look at the general information. That can be, first of all, who are you as a researcher? Which institute has the work been done? What materials have you been using? That's material information. I just shown chemical exposure, so you need to describe the chemicals. Which biological model did you use? So that's the next step. How is your study design? And then the readout. And what I did actually miss here, and that's not clearly indicated, is the raw data. I assume that's in this readout part, and then you have your analysis statistics for these final results. Now, once you have captured all of this, then where does it need to go? Well, I'll just give you one example, preferably on online repositories, and the why will come later. And I work a lot with the repositories from EMBL, EBI, and all the repositories are listed here. And the one that I use most is ArrayExpress here on the top for gene expression data, and also Biostudies, which will be topic in this webinar. And Biostudies, so that's the original paper from 2015. And nowadays, I already mentioned ArrayExpress, it has been fully incorporated in Biostudies as well. So you can go to one place and find all this information. And Biostudies has been used by quite a bunch of European projects and also now in the national project VHP4Safety. So all these projects have deposited their data in a collection in Biostudies. Marcia will talk more about Biostudies because she can nicely explain it in more detail, how that looks like and how it works. And the nice thing about this is you can still keep your data private until it's needed to publish them and make them public. So there's no need to already share them with everybody if you don't want to or need to. Then the next W is when. And when is basically as soon as possible. I've shown this already, right? So you know how your experiment looks like. You also want to know already before you do the experiment what type of samples you will collect and what further analysis you want to do. So all these aspects you can already describe. So that means don't wait until you got your final results. That's nice, but if you're already in data management or research output management, already start from the start, describing your experiment, write that down. Also your sample collection, what samples will I collect from my experiment, how I'm going to analyze them, create the raw data. And that would already be a point where you already have a nice overview of what you have done to deposit that in an online repository like Biostudies. Keep it still private. You do your data analysis, add that to that repository, and then also your final result in a paper, and you can link everything with unique identifiers. Then the last W, the why, well, it's to facilitate data sharing. And that's important for transparency of what you have done, and to share basically what your process is, how you got the data. It also increases impact of your own research, impact of other ones' research, and you can combine research. If you can use data from somebody else, use it with your own data, that increases the impact from both studies. And that's also then the part of the reusability of it. You already see this paper that nicely describes these elements, but also what then comes to mind is when you facilitate data sharing, that you also think about making your data fair, data, tools, anything that facilitates your research. So by sharing it, you make them findable, accessible, interoperable, and also the last part, reusable. And preferably then also when you publish it, make it open access so that everybody can use your data and your information and your result. Now the how, that would be actually 6W at the end, of course, of the how. And one of the examples, and this is something that Eliska will elaborate on, is a ToxTemp format, which is based on OECD guiding document 211. And that's basically a template for describing your cell-based toxicological test methods. So that it can be evaluated and also used for regulators. And that's actually my intro to the topic.
All right. Thank you, Daniel. Very nice. We have a Q&A session planned actually after Elishka's part, but there's already a question from Chris in the chat, so maybe I can just ask this one and then we move on.
Well, this question I can directly answer, and it's yes, but we can also elaborate more and later on.
Okay, all right. So shall we move on to Marsha then? Because I know that Marsha has to leave a bit early. So also if you have questions for Marsha, don't hesitate to put them in the Q&A, though she said Daniel should be able to answer all of them as well.
Can you see my screen?
Yes.
In the presenter mode?
Yes.
Perfect. So my name is Marsha Verheijen. I'm a postdoc at Maastricht University, and within ONTOX, I'm working on data management. So within data management in ONTOX, we actually have 2 data repositories, but today we will talk about Biostudies. We also have ONTOX Hub, but that is a bit different. So Biostudies is hosted by EBI, like Daniel already mentioned, and it's perfect for biological information. It can take any kind of files, so it can take Word files, it can take PDFs, Excels, but also raw files from analysis, so sequencing data, images, these kinds of things. And therefore, it's very perfect for biological data, while in ONTOX Hub, it's more like more structured data regarding chemicals and structures and these kinds of things. But we will not talk about ONTOX Hub today. So the data in biostudies can be kept private until publication. So that's a very, well, good point, because most researchers do not want to share their data until their paper is published. But from the data management point of view, as Daniel already mentioned, it's good to keep track of all your data the moment you start analyzing. So the moment you have data, it should already be in a data repository, because when you leave it for a year, you will forget details. And those details are crucial for reuse and FAIR. And another plus point of biostudies is that data accessions can be linked together. So you can have one data accession that actually has assay data, and you can have one accession that has ToxTemp information, and you can link these to each other so that they can always be findable together. So to go to Biostudies, you click on the link that you can see on the slide, and that brings you to the home page of the repository, which looks like this. So there's a lot of data already in the repositories, as you can see over here. And these are data accessions from many different projects, as Daniel already mentioned. And the publicly available data can be searched under the Browse button. So there you can see all the publicly available data that is in the biostudies. So it's 20 pages with almost 2 million results. But if you're going to search for data from your project, this might be a bit, well, chaotic. So you can filter actually on the project that you want to search on. So you do that in the collection. Of course, not all of the collections are represented on the main page. So if you need another one, you just click the See All. And here you can see the ONTOX project, which you then can select, and then you get data that is only from ONTOX. So currently we have 7 publications available. Well, actually, since this afternoon it's 8. We had a new release this afternoon. But of course, this is only the tip of the iceberg, because most of the data from ONTOX is still private and awaiting publication. So if you want to see the private data in the Biostudies repository, you have to log in. So all the ONTOX users have their login and can see the data. And then you can also find private accessions like the ToxTemp from Miliška. So you can see above the title that this one is still private, but there's also other information like the unique identifier of the entry, which is very important in data management because that's a unique number. If you search for this, you should always end up with this data accession. You can see that there's one link to the data file and 3 actual files to be downloaded. And the last modification of this document was March of this year. March of last year. And of course, every data accession has a title, which should be descriptive so that you can already have an idea of what is in the data. So just a title of raw data 1 will not really be useful. But when you click on the link, you actually go to the data page. So that brings you to a page like this. So on the left, you have all the details of the data, and these are free text fields, so we can actually put in whatever we want. And we have to make sure that it already describes the data a little bit, because only putting data files online will not be understandable for someone else in the future. So if you actually want to have the data files, you go to the right, where you can see which data files are uploaded, and there's a link to click download all. So it will download as a zip file. And of course, we also added a link to this page, and that's a link to a publication, which is important for this data. Of course, you're not always searching for a specific data set that you know that exists. Sometimes you might want to search for some liver-related disease to see if there's data available. So then you can use the search box. So I now put in a search for liver. And you can see that liver is highlighted in yellow all over the place. So in the title, but also in the summary. So if it's yellow, it's an exact match. You can also see green or peach highlighting when it's not an exact match, but a synonym or a more specific match. So that facilitates searching. Of course, you don't have to search in the ONTOX database. You can also search all of the other projects in case you want some data. So to get your data on the Biostudies, you have to contact your data manager. And you have to do this before you actually publish your data, before you publish your paper. Some researchers want to submit a paper and then come to us and say, we have data available. It should be the other way around, because every publication needs to have a data availability statement. The one we use in ONTOX is written out right here. So that means that the data has to be stored online already before, but it can still be private. For researchers, we can also make links so that only those specific researchers or reviewers can access the data. And also when your paper is accepted, you have to notify your data manager, because the data has to be made publicly available. But we also should add the link to the paper to the data accession so that all of the information is kept in one place. So don't forget to inform your data manager. And that's my short introduction on Biostudies. If you have questions, I would love to hear them.
Thank you, Marsha. Very interesting. I'm myself in the ONTOX project, and I've never actually really explored Biostudies because I tend to store my my own stuff on GitHub directly. I actually have the question: what would you say is the advantage of using Biostudies versus GitHub specifically for the data?
Yes, indeed. We also have GitHub in ONTOX. I keep forgetting about that. Well, GitHub is perfectly for scripts and these kind of and developments, while Biostudies is specifically made for well, free documents, so Excels and raw files, these kind of things. So perfect for analysis data. while structured information should actually go to the ONTOX hub. So all of them have their pros and cons. So if you're developing something, you go to GitHub. If you do lab tests, you go to Biostudies. And if you're doing PBPK, you would go to ONTOX hub.
And maybe I missed that part. I don't see any open questions right now, so I'm taking my chance. But maybe I missed that. But data versioning, how does that work in Biostudies if you have a new version of a data set?
Then we make a new accession number, and all the accession numbers are unique. So it gets a new number, and it can be linked to the old one to keep track of the different versions.
Okay.
You can also update existing ones if you need to add something, because we already had some created, but they were still missing part of the data, and that was later added. So then you get a new version, and you can always look back at the previous entries.
True, but like the physiological maps, we had a version 1, and then we got a version 2, so it's better to make a new accession.
It depends on what you're looking at. I know from one of Eliška's entries from a recent paper, data still needed to be added. Basically, the link to the I think that was the omics data, gene expression data, and that was added later as such.
I see there's a question of Chris in the Q&A. Why ONTOX Hub and not biomodels for PBPK?
That's a good question. I think we decided at the start of ONTOX that everything has to be present in ONTOX Hub. So it currently goes directly to ONTOX Hub. The ONTOX Hub itself will also have a link to GitHub and to Biostudies so that everything is findable. So that's our legacy of the project. I don't know why they didn't choose another run. I wasn't in that discussion.
All right. Okay. I don't see any more questions so far. So thanks, Marsha. Maybe we can move on to Eliška. Oh, wait, one thing. Is modified a type of versioning? Because I noticed there was a date provided with modified prefix.
There was a modified prefix.
I believe it is.
Yes, yes. When you modify an accession number, you get the new date of the modification, yes.
All right. Thank you. Eliška, I think the floor is yours now. We can't hear you.
Wait. Now can you hear me?
A bit faintly. So,
Okay, I will just switch off my headphones and switch them on again.
yeah, this is better.
Okay. Is it better now?
We can hear
Which
you.
one? I will just speak more loudly then.
Yeah, this is
better.
good. Yeah.
Okay, okay, perfect. I will share my screen. Okay, you should be able to see it.
Yes.
Well, so thank you so much for the invitation. And as Mari already introduced me, I'm a postdoc in the ONTOX project, and I work in the field of developmental neurotoxicology. And today I will share some user experience with the ToxTemps. But before I tell you why we have to use the ToxTemps, I want to introduce you to the topic itself, to DNT, which is short for developmental neurotoxicity. And if you are familiar with this field, at least a little bit, you might probably know that it's really bad that we don't know how toxic are chemicals to the developing brain. So if you imagine that there are hundreds of thousands of chemicals on the market, plus mixtures, then 99% of them were never tested for their DNT potential. And this is caused by the fact that those tests are not mandatory for developmental neurotoxicity, only after triggers. And if we test then these chemicals, we use only in vivo traditional guidelines like this OECD 426. And these guidelines use rodent tests, which means that they take very long, they are very expensive, and most importantly, the rodents are not really perfect for human situations. So the extrapolation of the results to the human situation is very difficult. And also it's not ethical if we have better alternatives, like the new approach methodologies that are in vitro or in silico assays that can predict the effects of chemicals on the developing brain. And so these NAMs, new approach methodologies in the DNT field, actually took a long time to be accepted by the OECD. So it took more than 20 years of really strong efforts of the whole DNT community, which then resulted in a document from the OECD published in 2023, which is called Initial Recommendations on Evaluation of Data from the Developmental Neurotoxicity in Vitro Testing Battery. And this in vitro testing battery contains 17 assays that assess key neurodevelopmental processes that you can see here on the image. It's, for example, proliferation of neuroprogenitor cells, their migration, differentiation into different effector cell types like neurons or oligodendrocytes. It can be synaptogenesis, myelination, etc. And if only any of these processes is impaired or disrupted, then the brain doesn't develop correctly. And we can then see, for example, cognitive impairment in children. And these assays, these 17 assays, they were developed especially by the teams of these three important DNT people. So Marcel Leis from the University of Constance, Ellen Fritche from the IUF, and Tim Schafer from the US EPA. And each of them have specific cell models, 2D or 3D, iPS-derived or primary cells, and each assess a different key neurodevelopmental endpoint. And so you see that in those 20 years, we finally reached the OECD document, and there were many mandatory and essential steps to reach this stage. And one of those steps to get the OECD adoption of our DNT NAMs was to have the methods properly described using ToxTemps. So of course, there were many other steps that had to be undertaken, but if you don't have your methods properly described, you will never get them regulatory accepted. And the ToxTemps of those 17 assays that I just mentioned, they are published as annexes to this OECD 2023 document. So if you want to have a look, if you want to see how they look like, get inspired, or while writing your own ToxTemps, just download them and have a look. And so these ToxTemps, what are they actually? So it's a template. It's an annotated toxicity test method template that was published first in this Krebs et al. Paper in 2019. You can also download it there. And it's very user-friendly, and it really helps you to describe your method properly. And so why you should use these templates? So these ToxTemp fulfill all requirements of the OECD guideline 211, as it was already mentioned by Daniel. So this guideline actually tells you what you should describe about your method, what are the mandatory information for other researchers or for regulators, risk assessors, what should we know about the method. These ToxTemps also guide the NAM developers in what information is required. That's what I said, that they are very user-friendly. They also include acceptance criteria for all the test elements, and they help you to define the cells sufficiently and transparently. So actually, all this information is necessary for other laboratories if they want to use your assay. So actually, it helps to fight the reproducibility crisis. So you probably already encountered, if you try to replicate an experiment of somebody else and you don't reach the same result, then the method is actually unusable. So if you have the method properly described, then it can be used by the whole community and also for regulatory purposes. The ToxTemp definitely built confidence in new approach methodologies, because if you understand what they can measure, if the risk assessors can see what they can measure, how the processes that they assess are regulated, if they know, for example, what are the applicability domains in the assays that are described in the ToxTemps, that definitely builds confidence into your NAMs. And you want people to trust your NAMs, to understand them and to use them. So in the end, the ToxTemps support the regulatory acceptance of your new approach methodologies. So as Marcia already mentioned, one of the ToxTemps is the NPC225 assay, as I said, as appendix to the OECD 2023. You can see it as a video on the right side. So it's one of our assays that uses neurospheres. It's a primary cell culture, and it's a multiplexed assay with multiple endpoints in it. It has 34 pages, so it actually took some time to write it. But if you already have all the information about your essay, just the copy-paste into the template doesn't actually take so much time. It could be, for example, 1 week of your work. But the development of the method, the characterization of your test system, that actually can take years. So what should be included in the ToxTemp? So of course, the test method description, the test system, meaning the in vitro model, also the test system source, how to get the cells if you want to use them yourself, the exposure scheme and endpoints. Well, usually you have the exposure scheme as a figure in your paper, so it's quite easy to paste it into ToxTemp as well. Then you have the handling details. This is actually quite tricky in the reproducibility crisis then. If you don't have all the details how to properly manipulate the cell model, for example, then you will never be able to get the same results. Of course, the data management, having proper metadata stored and raw data stored in Biostudies, for example. Then you have to describe the prediction model, toxicological application, publication validation status, method transferability, and safety and ethics. So most of these sections are quite easy to fill in. For example, for me personally, the prediction model is quite difficult. So for example, for our essays, we were collaborating with our mathematician that works in our team to help us create the prediction model actually. But for each of you, I guess other sections will be more difficult or easier. But as I said, it really helps you to summarize the information you have about your essay and put it in a structured way, and then easily share it with other researchers from the community. So I think it is very important to remember that ToxTemps are living documents. So my advice would be to start writing as soon as possible some first short version, and you can build upon it then when you have more data, when you have more characterization of your cell system, etc. But keep track of the changes you do, but start as soon as possible, because also by reading the ToxTemp, you will see the questions that they ask, and that will also make you think about what you should focus on, for example, in characterization of your cell model. So in our case, for this NPC225 assay, the version that was now published with the OECD document is our version number 2. And in the ONTOX project, we are already working in the next version. So this next version, just to give you an idea, will include characterization that is necessary for QIVIVE, so the quantitative in vitro to in vivo extrapolation. So we are right now measuring the protein and lipid content of our cell models. So this is done in collaboration with Wageningen University. So you can see on the slide Sylvia Adam, PhD student who did an internship in our lab in September, measuring the lipid content actually in our neurospheres, and her supervisor, Ninke Kramer. And we also focus on the metabolic activity of the test system, because if you expose your cell model to chemical, you want to know if they are able to metabolize the chemical, if the chemical can even reach the cells, etc. And another characterization that we are currently adding is the extension of the biological applicability domain. So here we actually submitted this week a publication on the regulation of key neurodevelopmental processes by neurodevelopmental disorders related signaling pathways. And this means you have the signaling pathways here in the middle ring. And what we did, we measured how they regulate the key neurodevelopmental processes, which are here. So the radioglia migration, for example, neuronal differentiation, how these signaling pathways regulate these key neurodevelopmental processes. Because this biological applicability domain then tells the researchers or the risk assessors what is the cell system able to measure. So for example, if a chemical will impact a certain signaling pathway that is outside of our applicability domain, then the cell system is not fit for purpose for this application. So this is very important to describe properly what is your new approach methodology able to assess. So to conclude, I hope that I persuaded you that ToxTemps are very useful and actually crucial to get your NAMs on the next level and to get them accepted. These ToxTemps are beneficial not only for the toxicology community, but also for you. And this is by the fact that these templates help you facilitate your new approach methodology description. It's very user-friendly. They help you to better understand your own method and the cell system. They help you raise questions about your new approach methodology, something that you haven't thought of before and that you should describe, should look into. And my advice is to start as soon as possible and keep track of the different versions and changes for transparency. And most importantly, the ToxTemps are beneficial for the toxicological community. You see the example I gave you with the DNT field. So first of all, they help fight the reproducibility crisis. They are essential for building confidence in your new approach methods and for their regulatory acceptance. And in the end, they also help you to replace the traditional in vivo toxicity methods with more robust, reliable, properly described and efficient human-relevant new approach methodologies. Well, this is all from my side, short insight into ToxTemps and their writing, and I thank you for your attention. If you have any questions, I will be happy to take them. And I hope you heard me properly then and that my headphones were okay. Was it okay?
Yeah, that was good.
Perfect.
Thank you, Eliška. I see there's two questions. Well, one is more of a comment from Chris, and I think it referred to the previous talk from Masha, that in general, project-based resources are less likely to be sustainable, that things like the Elixir deposition databases, including Biostudy, ArrayExpress, and biomodels, and these resources are also better known to non-toxicologists that might still have overlapping interests. So I agree. I think that was also a bit the underlying feeling from Marsha and Daniel. And another question that I think might be more related to your talk, Eliška, is we have seen time and time again that even if we think methods are described very well and it is not easy to reproduce a method and the results, do the projects also plan ring studies to evaluate that and/or exchanges to learn methods on site and extend protocols or ToxTemp itself based on lesson learned?
Okay, so the ring trials are actually very, very expensive, and usually academic projects simply don't have money for them. In the DNT field, though, there is a new project. I'm not sure if it's funded by EFSA. I don't want to give specificities, but all the DNT IVB methods are now being transferred to DNTox, which is a company. It's actually a spin-off of our team at the IUF. And this DNTox company is then gathering all the 17 assays, so from the 3 laboratories so they are now assessing the chemicals that were measured in the first EFSA project. These were around, well, between 100 and 200 chemicals, so we know which results we obtained at the 3 different laboratories. And so now DNTox is repeating those assays to see if we get the same results. So this is ongoing for the DNT IVB assays, and it's actually crucial that the assays are available for customers, for industry, for the risk assessors to being used, because if it's an academic laboratory, they can never assess hundreds of chemicals because they don't have time and money. So these assays have to be accessible at some company for purchasing. So not in the scope of the ONTOX project, but it is ongoing in the DNT field, just to make it short.
Thank you. I don't know if there's any other questions. You can also raise your hands if you have a question. Okay. Otherwise, I think we will move on to Jente, which will, if I'm not mistaken, propose a different perspective on ToxTemp. So I'm curious to see where the progress is there. Go for it.
I am now sharing my screen. Is that correct?
Yes, it's working.
Yes, perfect. So thank you, Eliška. For me, that was very interesting to hear your user experience with ToxTemp. So yeah, my name is Jente. I'm as Maddie also already introduced, I'm a data student and PhD student in VHP4Safety. And I would like to present on the ToxTempAssistant, which is a result from the third hackathon of VHP4Safety. which explored the use of large language model and their integration in toxicology workflows. So our problem statement was focused on a user story. So as you all know, researchers need to populate a lot of templates, including, for example, ToxTemp, and they're often faced with scattered or inconsistently formatted information that can be scattered across different sources. And we wanted to explore whether LLM can aid in this, so if they can effectively extract and structure the information. And we evaluated different approaches for their accuracy. So the template we focused on was ToxTemp, and I think Eliska already explained very nicely the aim of ToxTemp. So bottom line, support regulatory acceptance through the proper documentation of cell-based toxicity test methods. And therefore, because we are developing these in VHP4Safety, this is also a relevant template for us. We already looked at the structure of ToxTemp. It has 9 themes and about 77 questions. And I thought it was interesting that Eliska said it was quite an easy endeavor. It took her only 1 week. I also heard different sounds, that it's rather time consuming. But yeah, I'm wondering if what I'm proposing here is useful. So when we look at the structure, it contains many different topics, themes, 77 questions. And when we take a closer look at the questions, they are rather detailed. And because they are so varied, there are also several documents that can feed into ToxTemp. And I listed several here on the screen, such as scientific publications, SOPs, protocols, cell line characterization reports, maybe even data management plans, project proposals, etc. So why might LLMs be suitable for this task? So LLMs provide a novel way to semantically index and search data. And semantically indexing is organizing and retrieving information based on their meaning rather than by exact keyword matches. And this is not something new, but LLMs extends this beyond basic search and retrieval by offering also ways to summarize and structure the text. Of course, there are notable limitations, risk of inaccuracies, importance of oversight to ensure nuance and expert knowledge is not lost, and of course, the possibility of generating misleading information. So the key question here is, can we configure an LLM-based system to achieve a satisfactory performance and configure it in such a way that these benefits outweigh the potential limitations? So our approach doesn't necessarily focus on, what LLM to use, but rather on tackling the challenge of scattered data while maintaining the ToxTemp's quality standards for regulatory documentation. In this research, we only use two LLMs, so GPT 3.5 and 4.0 mini. And in this stage, benchmarking is not a priority. We rather focus on building a robust system. So following the hackathon, I continued to refine the large language model, moved away from GPT-4 to 3.5 to 4, which has a larger context window, which allows for broader contextual awareness in responding to the ToxTemp queries without the need for chopping up the document. So I try to configure it in such a way that the LLM adheres to consistent rules, encouraging really context-based answers and guiding the LLM to extract information based on relevant to the text test being documented. So the type of information you want to include is also a very short description of the test system that you want to describe with this specific ToxTemp to scope it. So how do we bring this scientific or this concept, this idea to the user? That is via web app development. We chose a modular and portable approach to ensure that it can effectively reach the end user. So it really serves the idea rather than the other way around. This is a simple process flow diagram. The idea is that the user uploads relevant context documents that are relevant for filling out the ToxTemp, including essay, which is essay name, which is incorrectly spelled here, and a very short description. And this description should contain the test purpose, the test system, and the endpoint. Then together with the prompt and one of the ToxTemp questions, this is fed to the serves as input for the LLM, which generates an answer or a draft answer, if you will. And this is done for each question individually. And that results in a draft-filled ToxTemp, which can be then be validated and edited by the user. And once you're satisfied, it can be exported to different formats. And this is what happens, might not be as relevant, but this is what happens behind the scenes. And now I would like to show the app. Hopefully it actually works. This is what it looks like, the ToxTempAssistant. You can log in with your ORCID ID. I now made up a mock investigation study. This is the essay. I already picked 6 files to upload on, and now I can continue to create a draft. Let's see if the internet allows it. Otherwise, I go to a pre-filled one. Let's see if it's working. It is working. Okay, it takes a while, so I can just go to a pre-filled one. Then you click on Edit Draft. So yeah, that's important to know. And this is what it currently looks like, which on the left side all the sections, and you can navigate through the different sections. You have the option, it's now in a markdown view, but you have the option here to view it, how it would be presented. There's a reference to the sources where this answer is retrieved from. There is a version history, which also summarizes the context documents that have been used in this first draft generation. And the idea is then that the researcher reads through the questions, maybe edits the questions, and once satisfied, clicks on accepted. So there has to be an active action to accept, or you have to read through the full thing. You can save it, and then you have a progress bar. Then there's also the option to regenerate a draft based on a single question or maybe on multiple questions. You can click on select updated questions, select questions for updates, and you again can upload a file. So yeah, it's a pretty simple idea. So once you're satisfied, you can export it to different formats, including JSON, but also a PDF, or this one might be the most readable. And then you get a filled out ToxTemp like this. It does clearly state that which GPT model was used, which ToxTemp was filled out, the base prompt. The JSON version also says LLM used yes or no, accepted yes or no, but this is yeah, a more final version. Yeah, and it clearly states when the answer is not found in the document, and that should also point researchers to where where information is missing. So yeah, that's to give you an idea of what the current system looks like. While doing this, one also realizes, okay, not all of these questions are suitable for LLM assistant population. Some already benefit from database linking. So that's also something I'm exploring. Then to the validation of the ToxTempAssistant. We of course need to know, does this actually solve the problem at hand? And to do that, I came up with a few tests to really understand the baseline capabilities of this system, including a positive control test in which you, as a context, you present a manually filled out ToxTemp template. So this is the optimal condition in which all the information should be present in the ToxTemp, and see how well it performs then. A negative control in which you provide something that has nothing to do with test methods or a certain test method. And then a robustness test that represents more real-life scenarios. Here are some examples of error types, such as factual omissions. For example, this was the human input, which listed the hairdryer, and while all the other special instrumentarium are listed, the hairdryer is not. Another one, for example, when there's the question asks for a database link specifically, the human input is CSOP, then the answer is not found because it's not a database link. And then we have over-answering or misplacement of information that is not necessarily wrong, but not in the correct spot. And one can discuss, okay, is this because the way the questions are written, or is this an actual error? Yeah, so in this stage, I'm trying to gather relevant context documents related to a single test method. And I'm trying to collect inputs from different organizations, projects, different endpoint test systems, ones that are more complex, ones that are more simple. And of course, different type of context documents, because some are, of course, way better structured than others. Then for the discussion, of course, we should wonder about the relevance of ToxTemp, or this specific version of ToxTemp. It's quite a flexible system, so it's just a matter of changing the questions, and yeah, then you could adapt it for different versions of ToxTemp. Of course, the verification of ToxTemps, the only thing this contributes to is to ToxTemps in general contribute to making the test more fair, but this, yeah, the ToxTemps mainly makes it more human readable. So for human reuse, it's not necessarily machine readable. Then we would have to go a bit further. And then, yeah, of course, this could help harmonize ToxTemp templates. And then for cost benefit, I think, or what I've seen and what I've also experienced, is that it's less mentally taxing to review a text that's already there rather than creating content, unless, of course, you can just copy paste it from another document. Then and that has already been reviewed. Then another point, there's already a EU tox risk database, and it would be nice to have interoperability with that database. And then lastly, the scientific merit of this exploration to what extent is this scientific, and does this contribute to the mission of regulatory acceptance of NAMs? And besides that, there are also several practical considerations, of course, especially legislative compliance when you use LLMs. The documents I use now were already publicly available, but I can imagine that there are more restraints when you might want to upload documents that have not been published yet. Yeah, that's it. Any questions?
All right, thanks, Jente. It's really cool to see how far this has come since the hackathon.
Yeah, you were also part of the team, of course.
Yeah, so it's really nice to see how this has evolved. My first immediate reaction would be to ask Eliška what she thinks of it, obviously.
Yeah, I'm curious.
Well, actually, I love your work, Jente. It's really amazing. I think it might save so much time. So, because I'm always searching into papers, where was this information published, then to copy paste it into the ToxTemp. So this would really save hours and hours of work. So is it already possible to use the app, or?
We're trying to get it released soon, but it's still being discussed where it will be hosted. And of course, there are several randvoorwaarden, preconditions that have to be thought about. But yeah, there's a lot of interest in it. So I think there's enough motivation to get it released as soon as possible. In this current stage, I try to involve researchers that then I generate a draft for them, and then I use their edited draft as an input to further refine the system.
Well, I will follow you and see when you publish, because I would really love to try it. I'm now writing another ToxTemp, and I would like to see the difference between what I write manually and what you have.
Yeah, that would be very interesting. Yeah.
Related to what you said about the hosting the app somewhere, so I think we had similar problem recently in our lab, because the mathematician in our team, he created a shiny app for data analysis, and we had actually the problem if we will share it on our servers, but then it would mean that the people actually share their data with us. So in the end, he somehow made it happen that it's always shared on their computer, so that they
Locally,
don't share the data, and then now it's publicly available and everything works fine. So maybe this would be your way, could be your way to go as well.
Yeah, I think the idea is now to Dockerize it, and then that you can use it locally indeed. But then there's still the LLM problem. Who's gonna pay for this?
Is it also in your plans then, once this is developed, to try to go to open source and self-hosting LLM or these kind of things?
Yeah, not sure yet. I need more input on that. Yeah, but these are all questions that are still open. Yeah. So
yeah, we have at the department some of the tools related to Visio and so on that are being hosted on a Strato server. So we need to look into that, how usable that is for this application, and the conditions that need to be met to do so. So that's still work in progress.
Okay. I see there is a question in the chat. I will encourage anyone who has a question to also put them here or raise their hands. The question is, does the ToxTempAssistant also import image and graphics, or only text?
I had this this question before, that there is people want to be able to upload images, and that should not be a problem. I now create a way to you can upload images as input, but not yet to place them in the text, because that would be nice, so you can immediately generate a full report, of course. But that should not be a problem, no, unless we have to save all of these images, then yeah, that's a lot of could be a lot of yeah, storage needed for that. So currently I'm not saving the documents, just the answers and edits of the answer.
And I think it's really good that you have this mandatory, review by the researcher that they
Yeah.
actually. Well, it can be that in the end you only click OK, accept, 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 but at least that there's this checking process. I think is really really necessary.
Yeah.
Do we have any other question for Jente or for any of the previous speakers?
No, it will be nice to meet with Eliska, see if you can.
I think you have another beta tester, Jente. All right.
So when I look on the Risk Hunter DB, your ToxTemps looked really nicely filled, but yeah, there's a lot of variation in the ones I saw for the for Risk Hunter. They released it now. You can download all the ToxTemps there. They filled out, and yeah, sometimes it really varies how they are filled out. And for me, it's hard to judge. Okay, is this sufficient or not? So yeah.
Yeah, that in general is an issue with filling out templates. Usually there's a minimum requirement. I mean that's what has been the case for the gene expression data, for instance. But what is minimum? And also fields could still be filled in with the ontologies are lacking in that sense, and that's a different ontology than we have an ONTOX, to be honest. So that's the vocabulary that's being used. So if you write human or Homo sapiens, still means the same, but the fields are too open, basically. And then if we can restrict that much more, then in this case with the ToX, then we have the chance to do that, to bring in this vocabulary more and more, so that it is really restricted to science, basically. And that will also then lead more to computer readability, right? Machine readability.
Yeah, exactly. But that would be that would definitely be a next step. I think first step is to get this a first version released.
Yeah. Eliska.
Yes, you were comparing the Risk Hunter and ONTOX-ToxTemps. I just wanted to say that the DNT ToxTemps, it's been years that people were working on them, because there are multiple versions, multiple people reviewed them, and years of experiments that gathered the data that went into ToxTemps. So that's why they are properly filled out. But it's understandable that any new ToxTemp then will take some time until it's the best way entered in. Yeah.
Any other question? Oh, Chris says the world would benefit from a general way to integrate ontologies in pull-down menus when filling templates, aka a software library and Excel functions.
Absolutely true. Yes.
Okay. Any other question or comments from the audience? Okay, I will. Annick, do you still want to close the conversation?
Yes. So I would like to thank everybody for joining us today. Thanks, of course, to all the speakers. Well, Marcia already left, but I hope maybe her colleagues can pass it on. And thanks to AlterTalks for their support in organizing this. And of course, thanks to everyone who attended the webinar. The next webinar will take place somewhere in the next 2 months, and it will be on stakeholder involvement. And for the early career researchers, well, please consider joining the SPIS Academy LinkedIn group, where you can stay updated about any webinars that they are organizing. And well, of course, you're also encouraged to follow the VHP4Safety and ONTOX channels for more information about events we're organizing.
Thank you very much.
Yes.
Have a good weekend, everyone. I think everyone deserved it now. And we'll see you at the next webinar.
Thank you, Maddie.